Hi, everyone. We are going to talk about hormones and sex today, and really going to be talking about our glands, right? So, how our glands work, how hormones work, and how, sexual development and gender identity unfold. And I want you to really notice how each builds from biology to behavior. And so, with that, let's start by talking about our glands. Now, first up, we have what's called excretin glands, and these are glands that actually send their products out through ducts. So if you think about sweat glands, think of salivary glands. It's anything that is releasing to the outside world or into a body cavity. Then we have our endocrine glands, and these are more private. They are actually releasing hormones directly into our bloodstream. Okay. And then we have our gonads, and our gonads are testes and ovaries. Okay. These actually belong to both the endocrine system and the reproductive system. And so they are producing those sex hormones like testosterone, estrogen, progesterone. And they do the absolute most. They're like multitaskers, and we'll talk a little bit about that. And then finally, we have sex chromosomes. And one thing to note about the X and Y chromosomes is it's not just about determining our biological sex, but they also influence hormone production quite a bit. The X chromosome specifically carries a lot of genetic information. So I believe there are more than 1,000 genes. And the Y chromosome is tiny, but it's mighty because the main job is turning on the processes that lead to the development of testes. When it comes to hormones, there are three classes of hormones overall. The first are amino acid derivatives, and these are basically hormones that are made from modified amino acids. They're working very fast. Examples could be epinephrine, right, which we know kicks in during stress. Then we have peptides and proteins. We've talked about this before in class, but these are chains of amino acids. And so some are short, some are long. They're some of the most common hormones that we have in our body, like insulin. Okay, they don't just kind of walk through cell membranes, but they need to actually bind to receptors on the outside, like they're ringing a doorbell. And then we have steroid hormones, and these are made from cholesterol. They can go right through cell membranes. They act more slowly, but they have really longer lasting effects. So if you think of steroids as kind of long-term project managers of the hormone world. And now I want to zoom in to three classes of gonadal hormones. And these are the hormones that are produced by the testes and the ovaries. Now, the first are androgens, um, testosterone essentially. And these are typically higher in males, but they are present in everyone. And they are involved in muscle mass. They're involved in libido and really development of the masculine characteristics. Then we have the progestins, progesterone, and these are especially important for preparing and maintaining pregnancy. But again, they do exist in all genders. So they are actually helping to regulate the menstrual cycle. They play roles in mood and behavior. And then we have estrogens or estradiol. And this is commonly associated with female development, but also it is very important in males as well because it is helping with our bone health, cardiovascular function, and really many aspects of brain development and mood. And so the big picture here on this slide that I want you to take away from is that hormones are really coming in different chemical forms, and gonadal hormones. And everyone has all three, but they have them in different ratios, so different levels. Now we're going to look at kind of the true VIP here of the endocrine system, and that is the pituitary gland. And we call this the master gland. And the reason why it's the master gland is because it's basically bossing around all of the other glands. It is releasing tropic hormones, which are really hormones whose sole job is telling other glands what to do. So if the thyroid, adrenal glands or the gonads are doing anything, the pituitary gland actually probably was the one that sent the memo for them to go do the things. Now, the pituitary actually has two parts, and they behave in two very very different ways. So we have the anterior pituitary, and this is really, you can think of it as the hard worker. It is producing and releasing a bunch of hormones on its own. And then you have, and things like that are growth hormones, right? So, like thyroid stimulating hormones, prolactin, it's very busy. And then you have the posterior pituitary, and this one doesn't make hormones itself. It is actually storing and releasing hormones that are made by the hypothalamus. So things like oxytocin or vasopressin. You can think of it as kind of storage and shipping. And speaking of the hypothalamus, the pituitary and the hypothalamus are like best friends. They are physically connected, and the hypothalamus is actually telling the pituitary when to release which hormones. And so together, they are regulating everything from stress to hunger and to sex hormones. Now, two big takeaways about gonadal hormones, which we've talked about here in the last few slides. The female hormones are actually cyclic, right? So estrogen and progesterone are rising and falling throughout the menstrual cycle. And this is creating very predictable patterns in things like fertility, mood, temperature, even energy levels. And then for males, they're steady. So testosterone levels do fluctuate, but not in a 28-day cycle like females, right? Men actually tend to have more stable day-to-day patterns, right? More like a slow daily rhythm instead of a monthly wave. Now, this illustration just shows a midline view of the posterior and the anterior pituitary and surrounding structures. And so this right here, and I will pull this out, is what we're looking at. So we have the anterior pituitary and the posterior pituitary. And if you remember, we have talked about posterior, post, behind, or in the back. The posterior pituitary sits behind this anterior pituitary. Okay. And the pituitary's anatomy and its links to the hypothalamus. Okay, so as I mentioned, the posterior pituitary is actually receiving direct neural input. The anterior relies on releasing hormones via the portal system. Okay. Now, let's talk a little bit about the posterior pituitary first. We'll break it down a little bit, but you can think of it as an extension of the hypothalamus. The hormones that are released here are actually made in the hypothalamus and specifically within the paraventricular nucleus and the supraoptic nucleus. And these neurons are actually sending their, hormone-filled axons straight down into the posterior pituitary, which is storing them and then releasing them. And there are two major hormones. There is vasopressin, which is also called ADH, and its main job is water balance. Okay. So the vasopressin, you can think of it as the hormone that is telling your kidneys, don't let all that water go, keep some of it. It is helping your body to reabsorb water and maintain proper hydration. The second one, the second major hormone is oxytocin. And oxytocin is very famous for its roles in uterine contractions during labor and even milk ejection during breastfeeding. It's also heavily involved in bonding and in social behavior. It is known as the connection hormone. So the posterior pituitary is really just releasing hormones that the hypothalamus has already made. Now let's go to the anterior pituitary, and it works differently, right? It doesn't get hormones sent down axons. It actually gets chemical instructions. And so we have hypothalamic releasing hormones, which send little chemical messengers like TRH, which we'll talk about in a little bit, and GnRH as an example, through a specialized blood vessel system. We have the hypothalamo-pituitary portal system, which is a dedicated shortcut between the hypothalamus and the anterior pituitary. And that ensures that hormones go directly to their target without getting diluted within the general bloodstream. And finally, we have hormones of the anterior pituitary. And once the anterior pituitary actually receives its chemical orders, it releases its own growth hormone. So things like TSH, LH, FSH, prolactin. We'll talk a little bit about that later. So, big takeaway here is the posterior pituitary is releasing hormones that are made by the hypothalamus. The anterior pituitary is making its own hormones, but only after getting instructions from the hypothalamus. So now we're going to move into one of the biggest breakthroughs in endocrinology, and that is the discovery that the hypothalamus controls the pituitary through tiny chemical messengers that are called releasing hormones. Now, before the 1950s and 60s, scientists knew that the hypothalamus and the pituitary were connected, but they didn't know how they were connected. And the big question at the time was, what is actually telling the pituitary when to release its hormones? And that's where the hypothalamic hormones come in. So these come in two flavors. There are releasing hormones, which tell the pituitary, hey, send out more. And then there are release inhibiting hormones, which tell the pituitary to like hold up and stop releasing. Now, these chemicals travel through the special portal system directly to the anterior pituitary to regulate hormone secretion. Now we have thyrotropin releasing hormone, TSH, from the anterior pituitary. I mentioned TSH, I believe, on the last slide, but the TSH will stimulate the thyroid gland to release its hormones. So if you hear the word or the acronym TRH or TSH, think of those as thyroid hormones. It is a perfect hormone relay race. And the reason why this discovery was so monumental is because it actually proved that the hypothalamus uses chemistry and not electricity to talk to the pituitary. And then we have the gonadotropin releasing hormone, otherwise known as GnRH. And that is one of the most important hormones for reproduction. The GnRH actually tells the pituitary to release FSH, which is actually known as the follicle stimulating hormone, and then LH, our luteinizing hormone. And together, they regulate sexual development, menstrual cycles, sperm production, and even fertility. Okay. Now, let's talk about different types of signals that regulate hormone release. How does the body control hormone levels? And one thing to know is hormones are not just floating around randomly. I know sometimes we think about that, and there are so many misconceptions that like hormones really control us and that they are our behavior is a direct result of just hormones, and that's just not the case. It's really a science. And I will say that your body really has an impressive system to keep things balanced. And there are three major mechanisms your body will use to regulate hormone release. The first actually come from neural signals. These are coming from our nervous system. So if you think of an example, you can think of stress, right? Stress activates our sympathetic nervous system, which then signals the adrenal glands to release adrenaline. Okay, this happens very fast. It's like flipping a switch. So you have the neural signals. Then you have hormonal signals. And this is when one hormone actually tells another gland to release its hormone. So you can think of it as, the endocrine system is sending its own chain of commands. Okay. And this is how most of the hypothalamus pituitary gland pathways work. And then you have non-hormonal signals, and these are other physical signals in the body that affect hormone release. Things like our glucose level, okay? Ion concentration, so like calcium, our stretch receptors, our nutrients in the bloodstream. A good example is the pancreas, right? The pancreas releases insulin when glucose rises. No other hormone was involved in that, just the sugar level itself. So now let's talk about the patterns of hormone release because hormones don't just come in one steady stream. There are a couple of things that we can talk about here. One is the pulsatile release, and many hormones are actually released in bursts or in pulses, and they're not released continuously. Okay, and GnRH, which I just talked about, is a perfect example of that. It has this pulse that controls reproductive function. And those pulses matter because if the pulse pattern changes, then the entire system reacts differently. And then we have minute to minute fluctuations. So other hormones in our body are adjusting constantly based on what our body needs right now in this moment. So cortisol, for example, has both the daily rhythms, but also has these rapid fluctuations during times of stress. Glucose regulating hormones, right? Shift minute to minute depending on what you eat. So, big picture here, hormone levels are regulated by nerves, by other hormones, or actual physical changes in our body. And the release is very rarely steady, right? It's responsive and it's constantly adjusting to keep us alive and to keep us functioning. So let's now kind of shift and look at how sexual differentiation happens during development and how the embryo really begins to become anatomically male or female. And the fascinating part about this is the default pathway is femaleness. So, in early development, every embryo actually starts with the same basic structures. And if nothing special happens, no extra signals, no specific gene activation, the body will naturally develop along the female pathway. And this is true regardless of chromosomes. Without additional instructions, the embryo will become female. Now, differentiation actually begins at around 6 weeks of gestation. So before 6 weeks, male and female embryos, they look the same internally, they look the same externally. And once we hit that 6 week mark, the body starts reading genetic signals that influence sexual development. And for males, the key is the SRY gene, and that's found on the Y chromosome. That is the switch that actually tells the embryo at that time to build testes. Now, if this gene activates, the testes will begin to form. And if it doesn't activate, even if the embryo has a Y chromosome, the body will typically follow the female pathway. Um, Internal reproductive ducts. So there are two systems. Every embryo actually starts with both sets of ducts. So there's the Wolffian system, which actually can develop into male reproductive structures, and the Mullerian system, which can develop into female reproductive structures. And only one system will continue depending on the hormones that are present. And then finally, by the third month, the testes will start to send signals. So if testes are formed because that SRY was activated, they will begin releasing two major substances. The first being androgens, which I talked about at the beginning. Those are the male sex hormones, especially testosterone. And they are promoting the development of those Wolffian or male ducts. And then there's the Mullerian inhibiting substance, MIS. And this is shutting down the Mullerian system so the female ducts don't develop. So basically, when we talk about male development, it requires two active signals: build male ducts and stop female ducts. Okay. And female development will happen if those signals do not occur. So let's look at how external reproductive organs develop, right? And again, the key idea is that all embryos are starting with the same basic structures. This is actually known as the bipotential precursor. And the reason why that is termed bipotential precursor is because it has the potential to become either male or female. Um, and when I say either male or female, I mean either male or female external genitalia, depending on those hormones. So everything begins from the same building blocks, and then testosterone or the lack of testosterone will actually guide the pathway. So let's go through each structure. First, you have glands. This is actually the rounded tip of the developing external tissues. And in males, it becomes the head of the penis, and in females, it becomes the clitoris. So when we're talking anatomically, the clitoris and the penis head actually come from the same exact embryonic tissue. Then you have your urethral folds, and these are folds of tissue that actually run along the underside of the embryo's developing genitalia. And in males, these folds will fuse together, it will close around the urethra, and will help to form the penis. And in females, they don't fuse, and instead they become the labia minora. Okay. And then third, we have the lateral bodies. And these are paired columns of tissue on the sides of the developing genitalial area. And in males, they're actually forming the shaft of the penis. And in females, they're forming the hood of the clitoris. And again, same starting material, different hormonal instructions. And then you have the labioscrotal swellings. And these are two swellings on either side of the urethral region. And in males, they fuse together, they form what's called the scrotum. And then in the females, they remain unfused and they become the labia majora. Okay. So, key takeaway from this with sexual differentiation, I would say, is each embryo is starting with the same blueprint, right? The presence of testosterone, especially the more potent form, drives the external functions and the structures that will develop the male pathway. Okay. Without those androgen signals, which I mentioned, the body will follow the default female pathway. And then the external structures will develop into the clitoris, the labia, and the vaginal opening. And this ensures that we're understanding sexual differentiation, hopefully, as a developmental process that are controlled by hormones rather than something that is predetermined from the very beginning. Okay. Now, let's shift into puberty, right? This is the stage where the endocrine system basically hits kind of that growth and transformation button. And puberty, what we know of it, and for those that have been through it, right, it is a massive biological change, right? And there are two major things that are happening here. The first is the growth spurt, right? And this is when our body suddenly shoots up in height. And the timing does differ between boys and girls, but the idea is the same. The body is accelerating growth very quickly for a short period of time. And then the second thing is the development of secondary sex characteristics. And these are the physical features that distinguish males from females, but aren't directly related or part of reproduction. So things like breast development in females, changes in voice pitch, menstruation beginning, body hair patterns, even changes in muscle and fat distribution, right? These are features that actually make someone look like an adult version of their sex, even though the reproductive organs themselves were built long before. And then we have the secondary sex characteristics. And these are not the internal reproductive structures, but they're everything else that emerges because of hormonal changes. Okay. And then the role of the hormones. So a few major hormones actually drive puberty. The first is growth hormone, and this one fuels the growth spurt. So bones, muscles, tissues all respond to it. Then you have your FSH and LH, which I talked about previously. Those are the gonadotropic hormones, and these are coming from the pituitary and it's stimulating the gonads, so the ovaries or the testes, which then will release sex hormones like estrogen, progesterone, and testosterone. And these sex hormones actually create very big visible changes during puberty. And then you have the adrenocorticotropic hormone, ACTH, and this stimulates the adrenal glands. These the adrenals will then, what will happen is it will release androgens that will contribute to things like pubic hair, underarm hair, some of maybe the early body odor changes that happen in puberty, even acne. So puberty really is a multi-hormone collaboration, right? It's not just, oh, testosterone in boys and then estrogen in girls. And puberty is the result of the brain, the pituitary glands, the gonads, and the adrenal glands that are all working together to transition the body into the adult form. Now, let's talk a little bit more about sex differences here. Researchers actually learned that the brain develops differently, and it develops differently based on the exposure to androgens like testosterone during specific windows of early development. So, androgen exposure at critical periods, let's talk about that a little bit. If the developing brain is exposed to androgens at the right time, it actually tends to follow a male typical developmental pattern. And if it's not exposed to these androgens, it will follow a female typical cyclic pattern, right? The one that supports the menstrual cycling. And this one is less about anatomy and just more about kind of the, I would say, organizational effects of hormones on brain circuits. Hormones are really just helping to set up the wiring. Now, androgen is, I mentioned the male pattern, and then no androgen is the female. Now, there's something called the aromatization hypothesis, and this is kind of the twist that surprises a lot of, I think, students. But testosterone actually has to be converted to estradiol to masculinize the brain. And that is what is known as aromatization. Now, it sounds backwards, but in many mammals, it's actually estradiol, not testosterone itself, that masculinizes certain brain regions. So why doesn't estradiol masculinize female brains? That is because in developing females, estradiol is actually bound by a protective protein called alpha fetoprotein. And it acts like a shield. It prevents estradiol from getting into the brain. So the female brain is actually protected from some of the effects of masculinity. And then testosterone bypasses this. It's not blocked by this alpha fetoprotein. And so in males, testosterone actually enters the brain. It gets converted into estradiol, and then that triggers, excuse me, the masculinization. Okay. So let's now bring this into the modern perspective. Because everything that I've just talked about is what is kind of covered in the classic model, but we know that science has evolved a lot, and we now understand that sex differences in the brain are much more complex than just testosterone during development. And so here's kind of what current research emphasizes, and again, continuous work in progress. But dimorphism is actually not simply a function of prenatal testosterone. And so there are sex differences in the brain that are influenced by multiple factors and not just one hormone at one moment in time. And these, include timing of hormone exposure, maybe sensitivity of cells to hormones, things like genetics, your environment, epigenetics. So brain sex differences are really complex. It's multifactorial. It's not one-dimensional. And then two, sex chromosomes also play a role. The brain isn't shaped only by hormones, especially the XX and the XY chromosomes themselves. Those influence brain development. So for example, some genes on the X chromosome may affect neural development and synaptic function. We've talked quite a bit about synaptic function. Your XX versus your XY cells actually behave differently, even without hormones. So some brain differences are showing up before the hormone surges occur. So chromosomes, hormones, they're working together and not in isolation. And then third, estradiol is important in brain development. And this is one of the most surprising, but also one of the most strong findings in kind of modern research, because estradiol, a form of estrogen, is a key player in brain development, but also across sexes. And it is affecting our neuron growth, synapse formation, brain plasticity, even things like cognitive development and mood and stress regulation. And in many mammals, estradiol is the actual mechanism through which testosterone, as I mentioned, masculinizes certain brain regions. So, estradiol we can just think of as not just the female hormone, but really it's the brain development hormone for everybody. So now let's shift from kind of the physical differences to the behavioral differences. So specifically, want to talk about how hormones during development can shape reproductive behaviors. And we know this also in studies with lab animals. So we'll talk a little bit about that. But most of what we know comes from experimental research, where scientists were really able to manipulate hormone levels at different developmental stages. And walking through some of the key findings. So first, reproductive behaviors in lab animals. Actually, researchers have found that exposure or lack of exposure to testosterone during early development can dramatically influence adult reproductive behaviors in animals. So, masculinizing females. So, when female animals are given testosterone during their critical developmental period, they may later show male typical reproductive behaviors as adults. And this is because there's been an early androgen exposure that helps organize the brain circuits that underlie those behaviors. We also have feminizing females. So, conversely, when male animals do not receive typical levels of testosterone during that critical development, they may display female typical reproductive behaviors. And so, the idea here is that hormones during early life will help organize the brain in a way that influences future behavior patterns. Now, there were early castration studies, and this research originally comes from, again, classic and yes, very ethically outdated by today's standards experiments where male animals were castrated early in life. And so, what was the outcome of that? Without early testosterone, their adult reproductive behavior resembled female typical patterns. Again, showing that testosterone is an organizing hormone, not just an activating one. And then we have perinatal testosterone and proceptive behaviors. Proceptive behaviors are the motivated or courtship behaviors that animals show when they are ready to mate. And studies show that testosterone exposure around birth, so the perinatal period, really influences, how much of those behaviors animals engage in later. So when we look at it from a hormone perspective, they're not just shaping anatomy, but really influencing motivational and behavioral patterns as well. Now, masculinization versus defeminization are independent processes, and this is a key takeaway. So masculinization equals developing male typical behaviors. Defeminization equals suppressing female typical behaviors. Feminization equals developing female typical behaviors, and demasculinization is suppressing male typical behaviors. Modern research actually shows that these processes operate totally independently. So, an animal can be masculinized in some behaviors, but not fully defeminized, or vice versa. Okay. So again, behaviors aren't simply male or female behaviors. It's a result of several developmental processes, each influenced by hormones at different times. So now that we've looked at animal research, let's talk about what this means for humans. I would say the first thing to emphasize here is that behavioral sex differences in humans are much more subtle and far more overlapping than in any animal species. So male and femaleness are not discrete categories. Human behavior doesn't fall into clean male or female boxes. Instead, I would say instead of thinking of behavior as two separate categories, it's really more accurate to imagine two overlapping bell curves with a lot of variation inside each group. And there's tremendous individual diversity, and people rarely will fit into these extremes. I would also say some behaviors are sexually dimorphic. So there are a handful of behaviors where average differences emerge across large populations. And so what are some examples? Examples often include certain spatial tasks, some verbal skills, maybe some patterns of play behavior in children. But even when these differences exist, they tend to be small and moderate, or small to moderate, excuse me. And there is huge overlap. So dimorphism doesn't mean all males do X and all females are doing Y. It just means on average, there may be trends, but that individual differences, right, vary widely. And most human behaviors overlap significantly. So for almost all of our psychological traits, personality, cognition, our emotional responses, our interests, the distributions overlap so much that you actually can't accurately predict behavior solely based on sex. And this is a key modern scientific point, right? Sex contributes to behavior, but it doesn't determine behavior. And hormones and brain differences interact with things like culture, entertainment, learning, experience, even social expectations, individual differences. And again, we come back to the fact that human behavior is multi-layered and it's not hormonally pre-programmed. And then finally, some differences in susceptibility to behavioral disorders. So where sex differences show up more strongly is actually in risk or prevalence of certain conditions. So for example, ADHD and autism are actually diagnosed more often in males. Depression and anxiety are more commonly diagnosed in females. Some disorders actually show different system or symptom patterns across different sexes. These differences also are believed to reflect interactions among things like our sex chromosomes, hormones, brain development, social and cultural factors. It's just not one simple cause. So, big takeaway from here is really human behavioral sex differences exist, but they're complex, they're overlapping, and they're shaped by both biology and environment. Now, there are otherwise kind of normal genetic males who suffer from what are called androgen insensitivity syndrome, which is they have normal levels of androgen, but their bodies lack the receptors to respond to it. And so, genetic males with androgen insensitivity frequently come to the attention of a physician when they become concerned about their inability to get pregnant, right? They're yeah, I mean that's often when it comes up is when you're going to see a doctor because you're having issues with this. And so after a physical examination, it may reveal that there's maybe sparse pubic hair, there's no menstruation, females have shallow vaginas, they may have internal testes and undeveloped male internal reproductive ducts. Their cells are all of the normal male XY chromosome types. And so during development, individuals with androgenic insensitivity syndrome develop along pre-programmed female lines, right, without the overriding effects of androgens. And in 1972, I think there were some researchers that found that childhood behaviors of these patients were those one would expect in a female in spite of their genetic maleness, right? They looked like females, they were raised like females, and they acted like females. So now let's look at the role of androgens. I kind of talked about, a physician diagnosing like males and females that have androgen insensitivity, but now we're looking at the role of androgens, especially testosterone, in male sexual behavior. So this slide is going to cover what happens when testosterone is removed, replaced, or just varies naturally. So first, androgens actually activate male sexual behavior. And in general, androgens are important for initiating and maintaining male typical sexual behaviors. And testosterone isn't the only factor, but it's a major activator. There's also something called an orchidectomy. And an orchidectomy is the surgical removal of the testes. And this eliminates, when that happens, it eliminates the major source of testosterone. So after removal, sexual motivation drops significantly for males. Erections maybe become rare or absent. Libido decreases, and these effects show that androgens are necessary for typical male sexual behavior in many species. The third is castration. So castration is just the broader term that includes the orchidectomy. And once testosterone production stops, physical changes actually can occur over time. So there may be reduced body hair, there may be softer skin, there may be a loss of muscle mass. And these happen because androgens also regulate many secondary male characteristics. And when testosterone is given back, right, it could be through implants, it could be through injections. Sexual motivation will typically return, and this shows that androgens have a direct activational effect on sexual drive. But the most, I would say, important nuance is that there's no simple relationship between testosterone levels and sexual motivation or performance. And this is one of the biggest misconceptions. And the reason why is because I would say after a certain minimum level of testosterone is present, more testosterone doesn't actually equal more sexual desire or better performance. So I would go back to say you could think of it like a threshold effect. If you're below the threshold, your sexual behavior, it will drop. But if you're at or above, adding more doesn't actually increase desire. So normal range testosterone levels don't strongly predict sexual motivation. And this is why. So men with high normal testosterone don't have higher libido than men with mid-normal levels. And even supplements or boosters will not magically increase sexual interest if levels were already normal. And the body doesn't respond to this like a simple kind of linear equation. It's much more regulated than that. Now let's go to the female sexual behavior and those hormones. So now we're comparing um, the behavior and the hormone cycles of females in rodents versus humans. And these two systems are actually taught very like side by side because rodents actually show very distinct hormone behavior patterns. While human patterns are much more complex and subtle. So it's nice to kind of look at it in lab animals. So let's start with rodents because most of what we know about hormonal control of sexual behavior comes from this animal research. And we'll talk about the estrus cycle. And so the cycle is about 4 days long in rodents, and sexual receptivity is tightly linked to hormone levels. And in rodents, when those hormone levels rise, behavior actually changes very predictably. Now, right before ovulation, the estrogen levels rise, and that typically makes females sexually receptive. So, behaviorally, they're ready to mate. And as that egg matures, what happens is there's this surge of progesterone, and this surge helps to prepare the body and is part of the pattern that leads to ovulation. And what is the fertility window? So in rodents, they're fertile for only 12 to 18 hours after ovulation, which is a very narrow window. And then there is something called an ovariectomy, which is the removal of ovaries. And when that happens, it completely abolishes the cycle. It will eliminate hormone-driven sexual receptivity, and this is because their reproductive behavior is directly controlled by estrogen and progesterone. And so in rodents, hormones and behavior, there's a very tight link. Now let's talk about humans, right? Humans are different. I mentioned we're much more complicated. There is actually no clear-cut sexual receptivity window, right? Human sexual behavior is not tightly tied to ovulation. It's not tightly tied to hormone spikes in the same way it is as rodents. And so while hormones are still fluctuating, behavior doesn't change in a simple, predictable way. Now, an ovariectomy in humans, if the ovaries are removed, menstrual cycling stops, fertility ends. Um, I would also say the most interesting part, though, of it is the ovariectomy does not eliminate sexual motivation in humans. So in other words, the libido is not solely driven by estrogen or progesterone. Okay, it's actually influenced by a lot of other factors, psychological factors, relationship factors, social context, stress, physical health, even personal history. So in humans, hormones matter, but they're not the primary driver of sexual desire. Now, I want to talk about the brain and sexual activity because this image is a cross section of the brain. Various parts are labeled, but there are four parts of the brain that are heavily related with sexual activity. And the first is the cortex, and the cortex actually right up here is going to mediate the most complex aspects of our sexual experiences. You have the ventral striatum, which is associated oftentimes with the anticipation and the experience of sexual activity, of other pleasurable activities, kind of like that reward, right? And then we have the hypothalamus right here. And the ventromedial nucleus plays a role in female sexual behavior. And the medial preoptic area plays a role in male sexual behavior. And then you have the amygdala, and that plays a role in the identification of potential mating partners. So those are the four parts of the brain that are really all about kind of sexual behavior. That those are the four parts of the brain that kind of helps to, teach us about the brain and sexual activity. So now I want to talk about sexual orientation a little bit because it is a complex human characteristic with, I would say, biological, genetic, hormonal, and environmental contributions. And there is no single cause of sexual orientation. Instead, research actually shows that it results from a combination of factors that interact during development. And so this slide really highlights a few important pieces of evidence. And the first is through genetics. So there have been studies with twins that give us some of the strongest clues. So concordance rates for identical twins are about 50%. So what does that mean? So if one identical twin is gay, the other twin is gay about half the time. And that's much higher than we see in fraternal twins or even in siblings, but it's not 100%. So, what does that tell us? That tells us that genes matter, but genes alone don't determine orientation. So, I would say sexual orientation is influenced by genetics, but is not genetically predetermined. And then you have early hormones. So most of what we know about hormones and orientation actually come from animal studies. And so we have to be cautious about applying those findings directly to humans, but they are useful for really understanding how early hormonal environments can shape later behavior. And so in animal studies, the perinatal castration, so in some male animals, if testosterone is removed very, very early in life, they sometimes show same-sex preference in adulthood. And this suggests that early hormones help organize certain sexual behaviors. And what about animal studies and testosterone treatment in females? So, similarly, giving female animals testosterone early in development can actually induce male typical behaviors, sometimes including same sex preference. And again, I would say these findings show hormones can influence sexual behavior patterns in animals, not that the same mechanisms directly determine human orientation. We also have the fraternal birth order effect, and this is one of the most consistent findings in research. Biological males with more older brothers are more likely to be gay. And this effect doesn't depend on social environment. It actually occurs even when the brothers weren't raised together. And so this tells us the effect is biological and not environmental. And then you have I guess I would say the one explanation of the fraternal birth order effect is maternal immune hypothesis. And so, what is this idea? So, this idea says that during each male pregnancy, a mother may build antibodies to certain male specific proteins. And so, with each successive male pregnancy, these antibodies may become stronger and could actually slightly influence sexual differentiation in the developing fetal brain. And this doesn't affect identity, it doesn't affect personality or masculinity. It's just one possible biological influence on orientation. And I will say, let's go here. Let's go to the next slide. So let's talk about sexual attraction. What triggers the development of sexual attraction? Sexual attraction is different from sexual behavior. And so what we're focusing on here is early developmental beginnings. So the stage when kids first start noticing others in a new way, which actually happens long before full puberty comes. And so this emerges around age 10 in many Western countries. And studies show that in Western populations, the first experiences of kind of romantic or sexual attraction, again, begins at around the age of 10. Sometimes it's a little bit earlier, sometimes it's a bit later. Now, this doesn't mean that puberty has fully started yet. Instead, it's just reflecting the first kind of stirrings of biological, of cognitive and emotional changes that lead into adolescence. And so different cultures and environments actually show different timelines, which tells us this is a mix of biology and social context. Okay. The key biological trigger here are the adrenal androgens. And so around ages 6 to 8, the adrenal glands start producing adrenal androgens, so like DHEA and DHES. And these hormones actually increase gradually and help kind of prime the brain for puberty. And so this is what influences mood and social interest and maybe early attraction patterns. And then by the age of 10, these adrenal hormones have actually risen enough to contribute to the first experiences of attraction. And then now I want to move into kind of going from biological sex and hormones to gender identity, which is related but also distinct. So, gender identity is really a psychological and social experience, and again, not just biological. So, first, I would say gender identity first refers to the internal sense of being a woman, a man, transgender, or another gender category. And it's essentially a person's deeply felt sense of who they are. And for most people, this aligns with their sex that's been assigned at birth, but not for everyone. And so gender identity often coincides with anatomical sex, but not always. So um, some individuals it doesn't align. And so this is simply just a natural variation in human development, right? Similar to how many traits exist on a spectrum. And this mismatch is not caused by parenting, by choices or socialization. Research actually suggests there's a combination of biological factors, hormonal influences, brain development, and even environmental contributors. And so again, complex and not reducible to one cause. Now, the term transgender describes individuals whose gender identity does not align with their sex assigned at birth. Being transgender is not a mental disorder. It is recognized as a human identity. And the psychological distress that some transgender individuals may experience is related to gender dysphoria, right? Not transgender identity itself. So, gender dysphoria, this refers specifically to the distress that someone feels when their gender identity doesn't match the anatomical sex or when they experience barriers to expressing their identity. So, a few important notes: not all transgender people experience gender dysphoria. Gender dysphoria is treated by supporting alignment between identity and lived expression and not by trying to change the identity itself. Professional guidelines really emphasize respect and support and mental health care that really affirms a person's well-being. And so, big picture here is that gender identity is really a deeply personal and central part of who someone is, and it's influenced by many factors: biological, psychological, social, and it's not determined by a single variable. So, this last slide is going to bring together kind of several concepts that we've covered. And I would say that sexual orientation and gender identity are separate aspects of who a person is. So while they can influence each other, they're not the same thing. Okay. So one does not determine the other. So biology defines sex characteristics like body type and reproductive anatomy, and gender identity is the psychological sense of self. Um, for transgender persons or transgender individuals, their gender identity does not match what their sex assigned at birth. And so their body type may therefore be inconsistent with their identity, especially before any transition steps. But their sexual orientation is completely separate. It's a completely separate part of who they are. So, an example of this could be a transgender woman, so someone who identifies as a woman but was assigned male at birth, may be attracted to men, women, both, or neither, just like any other woman. Okay, it's transgender man may be straight, gay, bisexual, etc. Okay, so gender identity is who you are, sexual orientation is who you're attracted to, and those two don't predict each other. Okay, so that is all I have for you today. And I hope that you learned a lot. We talked so much about our glands and hormones and how we develop as embryos, how we become male or female. We went through and we talked about the differences in males and females and the hormones and how that kind of impacts our behavior. And then we also kind of went through and talked about attractiveness, differences between males and females. We talked about puberty, and then now we're ending with sexual orientation and gender identity. So hopefully you learned a lot about hormones and sex today, and thank you so much for listening.